Good morning, everybody, and welcome to day 5 of our Evo week. So, over the past few days, we've covered so many different topics, but I think we're cohering around an understanding at multiple levels and from multiple directions on how organisms achieve the behavioral complexity that we see in nature through this process, combined processes of evolution and development. And today we're going to continue to explore these topics, but following Murray Shanahan's talk yesterday, continuing the theme of simulation, because one of the core ideas that underlies BCBT is that we can make the best progress to understanding nature by combining the sort of, if you like, the analytical sciences, particularly biology and neuroscience, which look at existing complex systems, animals and their brains, and try to figure out how they work, and we combine that with the synthetic sciences, where we start from scratch and we try and build something which is similar in an interesting way to that biological system. And following Valentina Breitenberg, who wrote a great book about this idea of simulating life, we think that the combination of analysis and synthesis gives us two very powerful tools to look at complex systems in nature and really get a good grasp on what's happening in them. And today we have two, I think, really interesting talks which will bring together ideas we've been looking at this week. Our first talk is from Randy Beer, who is in Indiana University in the US. I think I first came across Randy's work when I was doing my PhD, and his PhD had just been published as a book. And I think I was in my second year, and I read it, and I thought, wow, this is amazing. This is PhD work. It was very influential across the whole field at the time, and it made me much more ambitious in my own work, and also much more hopeful about the impact that a young scientist can have. And I think I wasn't the only person inspired by that book, and a whole generation, perhaps, of scientists have come into this field, and Randy is one of the leading figures. And so Randy's work is in simulation and modeling, but he looks at evolution, he looks at development, and he also thinks about this question of how organisms operate. It's not just about the brain, it's not just about the body, it's about the environment, all three things coming together and thinking about those as coupled dynamic systems. And I think this is the theoretical approach, which is really going to help us to unlock how complex behavior emerges. So Randy's been pursuing that approach. I think you were at Case Western, and then also involved with the Santa Fe Institute of Complexity. And so a lot of background and experience in both evolutionary systems and developmental systems. And so I'm really looking forward to this talk today. Randy.
Thank you, Tony. So I think I'm just going to jump right in. As Tony mentioned, I sort of take this perspective, and I have for a very long time, that understanding the mechanisms of behavior in animals is not about just looking at neural activity. It's not just about tracing neural connectivity. It's really a question about how nervous systems and bodies and environments interact as a coupled whole, and that's sort of the unit of analysis at which we need to work. I think a lot of us have been convinced of this for many years now, but I still have to sort of argue for it sometimes. I hope not here, and so I'm not going to, but this is still a very controversial position to some extent in some areas of neuroscience and in cognitive science, that bodies and environments and feedback through them is really all that important, and really it's the brain that does all the interesting things, and the rest is just sort of scaffolding around it. But like I said, I'm not going to talk about that today, so I hope for the most part, most people at least are willing to entertain that notion that there's this larger unit of analysis we need to think about. So when people think about brains or brain-body environment systems, it tends to be in terms of these grand theoretical schemes, which almost take ontological positions. So organisms are information processing systems. Organisms are dynamical systems. Organisms are prediction machines. Organisms are complex networks, and so on and so forth. Okay? That's not what I'm going to do. I think in general these grand theoretical schemes are not as productive as they could be, at least in their present form. It's really difficult to see how you would ever decide that one is right and the other one is wrong. It seems like they're all different sort of perspectives that one can take on a system, and they all have something to contribute to the conversation. So instead, what I want to emphasize today is I want to think instead in terms of sort of mathematical tools. So for example, there is a body of mathematics called dynamical systems theory, which we can think of as a kind of lens through which we can look at the operation of a system like a brain-body environment system. And when we look at that system through that lens, we can tell a story. We can come up with an explanatory narrative about how that system works in terms of the vocabulary of that mathematical theory. Now, that theory itself is not right or wrong, because it's not taking an ontological position on any scientific thing. Rather, dynamical systems theory as sort of a mathematical lens is just more or less useful for a particular purpose. And the nice thing about shifting to this notion of mathematical lenses is that there's no reason why you can't look through other mathematical lenses as well. So for example, I can look at the same brain-body environment system through the lens of information theory, for example. And I can produce an informational explanation about what's going on. And it's not the case that only one of those two stories can be right. In a complicated system, there are many different stories, each of which highlight certain aspects of a system. And what I'm particularly interested in these days is what's the relationship between these different explanations that arise when we apply different mathematical lenses to the same system. And so this slide sort of lays out what I'd like to accomplish here today. I want to talk a little bit about how one goes about modeling brain-body environment systems. I want to look at a particular brain-body environment system through the lens of dynamical systems theory, briefly, just to give you a flavor of what that's like. I want to then look at that exact same system through the lens of information theory, again, to give you a sense of what that looks like. And then I'd like to say something about how the two different explanations that those two lenses reveal might interrelate. Now, based on past experience, this is somewhat ambitious, so I'm going to get through all this in one talk. So the talk is somewhat modular, and also it can be driven somewhat by the audience. I understand that the way these things work is that lots of questions and so on. So that's great. I'm very happy to do that. And I'll just have to cut out different pieces of this story depending on how time works out. In particular, the order is as I just described. How do we produce these systems? How do we analyze them dynamically? How do we analyze them information theoretically? And then how do those two pieces relate? So if we run short on time, I may have to skimp on the relation bit. OK, so that's the sort of outline of what we're going to try to do.
One question.
Yep.
So you make a strong point here that you want to stay away from the very exclusive ontological commitments.
At least a priori.
Right.
Yeah.
But still you want to give us an explanation. So if you give us an explanation, you are making an ontological commitment, I would think.
No. I mean, the way I look at it, like I said, dynamical systems theory is a body of mathematics that's not right or wrong scientifically. It just exists. It has to be right mathematically, but in fact it is. By using those tools to describe any complicated system, there's a story you can tell about, through that lens, what's going on in that system. For dynamical systems, that might be in terms of attractors and bifurcations and transients and things like that. For information theory, it's going to be in terms of informational relationships between things.
Yeah, but you see now in your, I will not hassle you over this much longer, but you see now in your explanation or in your answer, you've switched to the notion of description. And this is maybe something we can inspect later, what we are achieving, whether we explain something, those are causal structures and causal interactions,
right,
or whether we're having, let's say, useful descriptions.
Yeah. OK. All right, so let's first talk about how do we produce these models. Now, it would be really nice, scientifically speaking, if, I think Eric Kandel said this, if there was an animal that had 3 neurons, they were big enough to see with the naked eye, pigmented, and you could basically read off their activity by looking at them, and that animal was conscious. Okay? It would be fantastic if there were simple animals that we could start with, just like in so many other sciences, we've started with very simple systems first and worked our way up to more complicated ones. But the fact is there are no simple biological systems. There are simpler biological systems, but even the simplest biological systems are very, very complicated to understand. And so a strategy that I've pursued for many years is to try to create, to synthesize, as Tony said, model brain-body environment systems that we can use as what in physics would be called toy models. Basically little crucibles of experimentation for thinking and probing different tools and different ideas as a way to develop possible theories, possible explanatory structures, possible bodies of mathematics, possible computational tools, and so on. And the particular approach that we've taken to that is by using an evolutionary algorithm. So the basic idea, there's nothing super clever here, it's a pretty standard evolutionary algorithm, is that we have some way to describe features of a neural circuit that, when decoded, is put into a body, and that body is put into an environment which undergoes behavior over an extended period of time, and then based on the relative success or failure of that behavior, that particular set of neural structures is given some assessment, some value, some fitness measure that says how well it did. And those different fitness measures are subject to a selection process and ultimately to mutation, crossover, and so on to produce a new population that once again is decoded, subjected to evaluation, and so on. Okay? And for us, I'm not so interested in this evolutionary process itself as I am in analyzing the results of this process. So I view this as a kind of, it's like our particle accelerator. It lets us smash nervous systems together until we get something that does something interesting, and then we want to study the operation of that system in lieu of being able to ask and answer the kinds of questions that we might ultimately like to ask and answer about complete biological animals. And I'll get back to that at the very end about how this can connect back up. Okay? So the thing I think that's most useful about using an evolutionary algorithm is ultimately these brain-body environment systems, these model brain-body environment systems that we're producing are selected not for our ability to understand them, not based on any a priori theoretical stance that we might take about how such a system must work in order to solve some particular problem that we pose. Rather, it lets us explore the space of possible solutions, possible brain-body environment systems that can exhibit a given behavior, that can be successful in a given environment. Okay? And because we don't sort of build in this a priori theoretical bias about how such a system ought to work, it becomes a very interesting exercise to examine through the lenses of various tools like dynamical systems theory and information theory, how these systems actually do work. Okay? And so that's the basic program that we followed for some time. And what I want to do is give you one example of this program, and then we'll use that example as a way to look at these analyses that I've been talking about. Okay, so the example is a relational categorization example. Relational categorization is to be distinguished from attribute-based categorization. So attribute-based categorization might say, this is blue, this is white. So it's a property of the object itself that we can use to sort all the blue things into one pile and all the white things into another pile, for example. We only need to look at properties or attributes of individual objects to do that. Relational categorization, on the other hand, involves relationships between things. So I might say that this is heavier than this. But heavier isn't an intrinsic property of this bottle of water. This is lighter than me. Okay? It's a relational property. And that's the kind of thing we want to look at. So some examples of relational properties are heavier, lighter than, larger than, smaller than, brighter than, dimmer than, things like that. Okay? In particular, we're going to look at a smaller than relationship. And the task is basically going to be set up as follows. We have an agent, a simulated agent that can move back and forth in its environment. It has an array of distal sensors, and objects drop towards this agent from above. And the basic task looks like this. There's various ways you can do this, but this is the one I'm going to focus on today. So there are two stages. In the Q stage, what happens is an object of some size is dropped, and the agent can do whatever it wants. We don't care. We're not going to pay any attention to what it does for the purposes of evaluating this agent. Eventually, the object reaches the agent, and the object's removed from the environment. Then a second object is dropped. And here's where what the agent does matters. Okay? If that second object is smaller than the first object, the agent is supposed to catch it. If the second object is, on the other hand, larger than the first object, the agent is supposed to avoid it. So it's expressing its relational discrimination between the second object and the first object through its behavior of either catching or avoiding, rather than just turning a light on if it's in the smaller than category and off if it's in the larger than or something like that. Okay? So the actual agent that solves this problem is quite simple. It has 7 sensory neurons, each of which is related to one of those rays that you see sticking up. It has 3 interneurons, which are fully interconnected, and it has 2 motor neurons, which collectively push the agent one way or the other, depending upon the balance of force being produced. I've mostly tried to avoid equations here, so let me just tell you the important thing is the model neuron that we're using is dynamical. So even a single neuron has some temporal response associated with it in general. It's not just an input-output device. And so this nervous system, as simple as it is still a dynamical nervous system. Outputs change over time of the neurons, even if you put constant inputs into the system. We can talk more about those details if people are interested. But again, I think it's a largely uninteresting piece of this, the particular model that we use here. One other thing I need to tell you is that if you do multiple evolutionary runs on this setup, you get at least 2 different kinds of solutions, which I'm going to call passive agents and active agents. Passive agents just sit still while the first object falls. They don't do anything. They passively observe it. And then when the second object starts to fall, of course they have to move because their decision is expressed through movement. Okay? In contrast, the active agents are always moving. Even when the first object is falling, they're moving around in particular ways. Okay? Interestingly, this is a distinction that wouldn't make any sense if this agent weren't actually embodied, even in this simple way. If we just had an abstract categorization neuron or something, then this notion of passive versus active strategies wouldn't actually be allowed. So embodiment and situatedness raise new possibilities like active perception that might not otherwise be possible.
So Randy, for clarification,
yep.
What are the kind of categories that you expect to come out of this? So how would you label those?
Well, the way the task has been set up, we would call these smaller than or not smaller than, which you might call larger than or larger than and equal to.
Okay, but what distinguishes this from classification? Like, classification is really that you are sort of processing the sensor data itself and cluster it in some way, while categorization means you're imposing almost arbitrary boundaries among these clusters, right? So for instance, I can say we have humans as a category, and then I can include a doll at some point in this category when I wish to do that.
I'm not going to argue about words. This is an example of what in cognitive science is called categorical perception. If you actually look in the literature, for example, there are examples of this in insects. You can see this in rodents. You can see it in pigeons and so on. And it's called relational categorization in those areas.
Well, we can debate that because actually in insects, this would be, let's say, a looming response, right? You would look at avoidance responses triggered by a looming stimulus, on which we have been doing quite some modeling. In that literature, this would never be described as categorization, right?
I can't comment on that literature, but there's certainly in the psychological literature, the word categorization is used in this context. But I'm not going to argue over the word. The fact is, you're breaking what is really a continuum of possibilities, namely all possible sizes of these two objects, and you're carving it into these two discrete sets, smaller than and not smaller than. Okay? All right, so the first step then that I want to do is talk a little bit about how we might think about so let me see, I don't know if I got to this or not. So we've got the passive and active agents. I'm going to focus on the passive agent initially, and I'm going to return and talk about the active agent later. Okay? The passive agent story is simpler, and it forms a nice background against which we can talk about the active agents. So let's first look at this passive agent through the lens of dynamical systems theory. That is, in terms of the concepts of dynamics. So the way we've always done that is as follows. The simplest kind of decomposition you can imagine in this system is that there is an agent and there is an environment, and each of those have some dynamics associated with it, which I've just represented here as sets of differential equations. So there's some set of state variables for each. There's some laws as to how those state variables evolve in time, and there's coupling. So the sensory inputs to the agent S come from some projection, some function of some of the states of the environment. And the motor inputs, if you will, to the environment come from some subset of the states of the agent. Okay? So that's all you need to read from that. So there are two, in dynamical systems theory, there are at least two different ways we can think about this system. On the one hand, we can think about it purely autonomously. And autonomous is a technical term in dynamical systems theory, which is only somewhat related to its use in, for example, autonomous agents or autonomous robots. Autonomous basically means that all the laws are given by the dynamical system itself. There's no external time-varying inputs coming into the system. Okay? So if we have this coupled agent environment system, with nothing outside of it, then basically we can collapse the agent variables and the environment variables into a unified set of state variables, and the agent and environment dynamical laws into a unified set of dynamical laws, and we end up with an autonomous dynamical system, which the tools for analyzing are extremely well developed, and we can do things like, I guess maybe you've already heard about some of these things, so we can identify the attractors. In general, we can identify the limit sets of this dynamical system. We can characterize their stabilities. We can look at bifurcations of these limit sets as various parameters of the system are varied, and so on. So this is a very highly developed body of mathematics that's it's about 125 years old at this point. On the other hand, we can pull these two systems apart and think about the environment and the agent as distinct dynamical systems, which are being driven by time-varying inputs. Now each of these systems are non-autonomous dynamical systems. And we can ask questions about how the dynamics of the environment, for example, changes as we manipulate these motor inputs to the environment, right? What's output to the agent becomes input for the environment, and vice versa. Now, the problem here is that the mathematics of non-autonomous dynamical systems is not nearly as well developed as it is in the autonomous case. And so we use, and then the trick is to relate these two. Okay? So what we use is something called quasi-static approximation. It's also called adiabatic approximation in physics, where basically what we do is we replace. No, I'm just looking. My mouse ought to work too, but I don't see it showing up. Well, we also have a pointer. All right, now I have it here. I have it here. So what we do is replace these time-varying sensory and motor inputs with fixed parameters, S and M, that we can vary. And that turns each of these back into an autonomous dynamical system that we can study as a function of those two parameters. So to give you a very simple example, this colored portion is just a particular way of representing how a very simple agent dynamics might vary as a function of the sensory parameter. And then we can go from this sort of quasi-static understanding to say, well, what happens if this parameter S, in this case there's just a single one, starts varying slowly in time? Okay? And then as it varies faster and faster, we can trace out the actual trajectory and relate it to the underlying autonomous dynamics of the system. And it turns out that you can prove that if this varies arbitrarily slowly, the dynamics of this system and the dynamics of this system are going to be the same. And as this starts varying faster and faster, the dynamics of this system and the dynamics of this system are going to become more and more different. So what this lets us do is use this sort of autonomous dynamical ideas when the time scales are not too badly separated in order to understand something about this non-autonomous dynamical system. And that's kind of the game that we have to play. We want to understand this guy, our coupled brain-body environment system, either in terms of this analysis or this analysis. And this is actually the one I'm going to be talking about today, the non-autonomous case, which is why I spent the time to talk about it. Now, one point I want to make is I've just said all of this about brain-body environment systems. But in fact, there's lots of different ways that you might want to decompose a dynamical system. So you can look, you can do not just an agent environment, but you can subdivide the agent into the nervous system and the body. You can look at this agent environment dynamics, as we were just talking about. You can look at neuromechanical dynamics, which is a very big issue in motor control, where you're looking at how the body dynamics in interaction with the neural dynamics produces a particular behavioral response. You can subdivide parts of a neural circuit to look at the internal dynamics of a neural circuit. You can also partition up the system in other ways. So for example, you're asking about multi-agent interactions, where each component now is going to be an agent, and we're interested in the dynamics of the coupled interaction of all the agents. All of these things can be easily formulated in the way that I just did. So it's in some sense mathematically a very, very similar problem. Okay, so a little bit of abstraction. Let's get back to our agent. So just to remind you, there are two stages. The Q stage is that first object that we drop that sets the baseline for comparison. And then the probe stage is the second object we drop that you actually catch or avoid. So this is, I want to first talk about the Q stage, that initial object. So, and just also to remind you, this is our circuit, and the picture I'm going to be drawing here is going to be focused on the interneuron dynamics, okay, which I've highlighted in red there. So let me throw up a couple of pictures for you here. This is the way you want to think about it. So this is a non-autonomous dynamics because it's being driven by these time-varying changes of the dropping object. Okay, so the sensory inputs are changing from moment to moment. So if we look at the dynamics of this set of interneurons, which we can plot in 3 dimensions, that's all I'm doing here, N1, N2, and N3 are just neuron 1, 2, and 3 here. At any given point in time, the state of the system is at some place, and that's what I'm going to show you in brown. Okay, so that's where the state currently is. At that point in time, if the sensory inputs didn't change, then there's some attractors in the state space, which I'm showing you here in blue, which the state is being pulled toward under the dynamics. Okay? And then the connection between them is just showing you the trajectory that the state would take to that attractor if nothing changed. Of course, things are changing, though, because this object is falling. So the sensors are changing in time. And I'm going to show you movies as to how that all plays out. I'm going to first show you one for a smaller Q object, and then a movie for a larger Q object. So let's first focus on here. So this was where we started. The brown is the actual path that it followed. And as you kept seeing things appear and disappear, the autonomous dynamics of the circuit were undergoing bifurcations. And the state is constantly chasing this. So there's an equilibrium point. There's an attractor over here. So I'm moving in this direction. Oop, that disappears. Now there's one over here. So I'm moving in this direction. I'm transiently chasing these moving attractors. That's a picture of that non-autonomous quasi-static dynamics in action. And at the end, when this object has fallen to the level of the agent, I get to a certain point right here. Let's look at the same thing now for a larger object with a size of 50. So the autonomous dynamics are a little bit different here. It's hard to compare it unless I played the two movies at once, but there's somewhat different sequence of bifurcations, and the equilibrium points occur in different places because you're getting sequences of different sensory inputs as a small object falls versus a large object. Yeah? The green dots are saddle points, if you know. Yes? Does that make sense? Okay. Yeah. So it's just a different stability for people that don't know what that is. But it's mostly the attractors that we care about, which are the blue points. Okay? One other comment to make here is that notice that the state you end up in when you have a smaller object is somewhat different from the state you end up in when you have a larger object. So the next thing I want to focus on is a movie of how all that fits together, how these different paths that you follow over time, this transient dynamics, depends on the size of the object. So I'm going to show you the same. We always start in the same place, but we're going to evolve somewhat differently based on the size of the object. So this is just how the dynamics of the system unfolds over time for many, many different sized Q objects. Okay? And at the end of the Q stage, we have this curve, which is a curve along which you're going to end up depending upon the size of the object that you encountered. And it actually turns out to be very nicely ordered. So the largest objects leave you at the top of this curve, and then smaller and smaller and smaller objects, you move down this curve until the smallest objects are down at this end. Okay?
So N3 is then your main dimension on which this is organized.
It's actually organized along all three dimensions, but it looks from this projection at least that N3 is the most important one.
That's your one interneuron.
There are three interneurons.
Oh, right, but N3 is one of them.
N3 is one of them. Yeah, this whole plot is interneuron activation space. Now, again, I want to emphasize that this is showing you the result of one evolutionary run. Different evolutionary runs, of course, are going to produce different sequences of bifurcations. They're going to produce different paths like this. But they always have the property that at the end of the Q stage, there's some structure in state space. It's not always quite so straight. It's not always aligned more with N3 than N1 and N2. But there's always a structure in state space that's organized according to the size of the first object based on this transient dynamics that I've just described to you.
Yeah, so, but Randy, I was making that point because I know, of course, you just want to have an example that we can all understand. Mm-hmm. But in some sense, if you have a single interneuron that does most of the work here to classify between small and big, then it's a very reduced dynamics that you're looking at.
It is. Well, and in general, I mean, if you think about it, we have a three-dimensional state space here, and we have one parameter, right? And so it's going to be a one-dimensional curve, right? So it's always going to be a reduced description. But it may not always be so nicely laid out. There's nothing that says the curve couldn't be looped around in some way in three-dimensional space. And sometimes it is. But it's always going to be a one-dimensional description by the nature of the setup. OK. So the next thing I want to point out is there's a bunch of interesting features. I mean, I've shown you that using this sort of quasi-static thing, you can explain each property of this entire, why it goes down and bends back up and so on. All of that is a matter of chasing these different equilibrium points, and all that can be worked out in great detail. But the important things for us are simply that, as I said, you have this curve. And the other interesting thing is notice there are some really sharp changes in direction, like this one, for example. And we saw in the quasi-static story that that's places where bifurcations occur. Okay, so you're chasing one equilibrium point, and suddenly it disappears, and now you're chasing another one. You change direction. Where does that come from? Well, if we look at the geometry of the situation, as objects fall in the field of view of this agent, there are particular places where a ray that wasn't intersecting an object suddenly intersects it, or a ray that was intersecting an object stops intersecting it. Okay? So this is like having, only 7 pixels in your vision. Okay? It's going to be a really important event when one of those pixels becomes occluded or unoccluded. And again, I'm not going to go through the details, but we can calculate the time at which an object falling at a given speed will intersect each of those places, each of those rays, and we can calculate from that where you would be in the interneuron state space trajectories at that time. And what you'll find is that these 3 points of intersection correspond to these 3 curves in state space, which are exactly where those discontinuities occur. So in this agent, what's causing the bifurcations in the dynamics are basically the making and breaking, which are discrete events, of ray intersections in its field of view. Okay? All right, now what I want to do is talk about the second object, the probe stage. So now we've dropped one object. Just to orient you, remember at the end of the Q stage, we're somewhere along this curve, ordered by large to small. Okay? Now that object goes away, we drop a second object, and what I've shown you here is a set of trajectories colored according to whether or not the probe object, the second object, is smaller than the Q object, in which case I've colored it blue, or larger than the Q object, in which case I've colored it red. And what you see is is generally what you would need to see for any agent that's making a discrimination. There's got to be some divergence, right? So although they start out all mixed up, I'm showing you for 5 distinct Q size objects, what eventually happens is they very sharply separate into the blue and the red trajectories. That's what a discrimination looks like in the interneuronal state space of this agent. Okay? So that's what we need to explain. That's what we need to understand dynamically if we want to understand the dynamical basis of this discrimination. So I'm going to do that in 2 brief steps. First of all, I want to look at the very beginning. What's going on right up here? Okay? So I'm just going to zoom in on that part. Is redrawn over here. Here's that curve at the end of the Q stage. Here are several different bundles of trajectories at different Q sizes. And what happens is this one-dimensional curve now spreads into a 2-dimensional shape, which depends upon the size of the Q object and the size of the probe object. So this curve, this one-dimensional curve, now smears out into a sheet, which again makes perfect sense because you've got 2 independently varying parameters now, the size of the Q object and the size of the probe object. That's going to describe a surface, a 2-dimensional geometry embedded in this 3-dimensional interneuronal state space. The other thing I've shown you here is something I'm kind of looking forward into the future. I've colored the parts of this 2-dimensional manifold blue that will eventually be caught because the second object is smaller than the first. And I've colored red the parts which will eventually develop into avoidance because the second object is larger than the first. Notice that they haven't pulled apart yet. These are slices through what's actually a continuous 2-dimensional object in the state space. Okay? But we've seen a spreading out. Now, what actually pulls this apart into these large differences of behavior? Well, that turns out to be nicely illustrated here. So the important thing to see here is the following. I've picked one Q size, and I've dropped a whole bunch of different probe size objects. Okay? And some of them I've colored blue because eventually they pull over here like this to be caught. and some of them are colored red because eventually they pull over here to be caught. So this is just a projection in N1, N3 of this picture. Okay? The key thing that happened here is very similar to what happened in the transient dynamics of the probe stage. A bifurcation occurs so that now there are two different stable equilibrium points. And one of the most important things that saddle points, which someone asked about earlier, these green things do, is they tend to be associated with the boundary between a basin of attraction of one limit set and the basin of attraction of another one. So if you're on one side of the boundary, you're going to go to this equilibrium point. If you're on another side of the boundary, you're going to go to this equilibrium point. And it's the combination of this spreading out of the curve into a two-dimensional sheet and then the bifurcation that occurs in the dynamics of the system that ultimately pull this apart into a catch versus an avoid response. And this is just another picture. So this was a movie I showed you in time. This time here is this axis. So you can see that as these bundle of states evolve, they eventually start to pull apart a little bit because of this effect. And then this bifurcation occurs, and suddenly there's a boundary such that you're going to be attracted one way or the other way, and that splits them apart. Okay? So the story that we basically have here, the dynamical story, is that the first object, because of the different non-autonomous dynamics that you get from falling objects of different sizes, the first object spreads the possible states out into a curve, a one-dimensional object in the interneuronal state space. Okay? Then when you drop the second object, that curve gets pulled into a sheet, a surface, which is then split via a non-autonomous bifurcation in the interneuronal dynamics, causing it to diverge into these two bundles of catch versus avoid. That's a sort of quick summary of the dynamical story that you see in this passive agent. Now, as I said before, the details of that story are going to be different every time. But the general way I just described it actually holds true for many, many, many passive agents. It's just which particular curve they spread out on, which particular surface they spread out on, and exactly how the bifurcation underlying it slices that surface into two pieces that then diverge. Those details differ from one agent to the other to the other. But the general form of that dynamical story as to what's going on in these agents holds across these passive agents. Okay, that was a quick overview of sort of how one would go about doing a dynamical analysis of one of these agents.
Before we move to the information theory,
yep.
One thing that confuses me, I mean, the strength of this sort of view of dynamical systems for these examples is that you can approach it analytically, right? So when people analyze these kinds of models, you go to some master equation that helps you to understand how an energy landscape that is a macroscopic description of dynamics evolves, and then you can derive all these properties analytically. Right? But in some sense, you're going through this now in a sort of an experimental way. You try to reveal this through experimentation.
I have to disagree with several of the things you just said. First of all, there is no potential function here. In fact, potential functions are quite limited. They really only apply to equilibrium points. And although this agent only has equilibrium points, it's quite common to see limit cycles appear as well, which can't be as directly captured by potential functions. Of course, chaos also can occur and is completely uncapturable by
what we look at here, what we look at here could be captured.
Yes, this right? Could be captured, although I'm not sure it's the most. I mean,
me neither. I don't know either, but that's why I'm asking you, right?
There's no master equation approach here, so this was mostly done numerically. Because what happens is so this is a common, this is worth talking a little bit about. This is a common question that people have. There is a body of mathematics, dynamical systems theory, which you can exactly write out the underlying equations that determine these various things, okay? And so in that sense, it's all mathematically rigorous. On the other hand, for almost any interesting system, including these, you can't solve those mathematical equations that you write down. And so you end up solving them numerically, which is not the same thing as you couldn't write them down in the first place. It's not the same thing as just running a bunch of simulations and figuring out what happens. There's this intermediate step, which is usually where we live in all the work that we do, where you can write them down. They're rigorously defined mathematically, but to solve them, you have to resort to numerical techniques.
Okay. But now, if you take the perspective on this task of, let's say, a locust LGMD neuron, right? It does the looming detection and the collision avoidance. So that means if an object is approaching at a certain velocity, it will trigger at some point an escape reaction. That's the same task, right, that's being performed here. In that case, what this neuron is exquisitely tuned to is angular velocity and angular size, also the dynamics of the input.
Mm-hmm.
Right? Well, here you seem to focus more on, let's say, a static detection of size with a sort of a thresholding approach. So, would that, if you would like to scale this up to the dynamics of your input?
I mean, there is dynamics here in that, for example, this wouldn't work. Okay. So there's a detail here which I've kind of glossed over, which maybe I should talk about. If you notice this trajectory right here, it's actually blue, but it's on the wrong side of the threshold line. Does everybody see that? Right? It doesn't show up very well on the projector, but this is blue, okay? So if it were just a static thing, then this makes no sense. And the reason is it's not a static thing. That's why I showed you this movie. If you watch, this moves. This boundary itself is moving in time, and the trajectories are moving in time, and it's actually kind of a race, right? The trajectories are moving and the boundary is moving, and it's a matter of who, at the end of that result, some trajectories are left on one side and some are left on the other side, either because of their own movement or because of the moving boundary. And it's a dynamic effect that actually slices this in the right place. Okay? I agree with you that because of the nature of this particular task, it is making a categorical decision, which is a kind of a discrete decision in the end. Okay? But there's actually nothing about the evolutionary approach or the way I've analyzed this which assumes that. And you already see that in the fine details there are dynamical effects. And if this were a problem that required those dynamical kinds of judgments, like velocity judgments rather than just size judgments, which is what this is about, then you would see that play a much bigger role.
Okay.
Yes?
So does what you just said imply that it's going to be sensitive to falling velocity?
Yes, it is. It is, yeah. And in fact, in this particular model, that was fixed. What tends to happen is the things you want it to generalize over, you need to vary during evolution. So if there's any feature of the model that you hold fixed during evolution, then typically the evolved brain-body environment system is going to latch onto that feature and assume it. If you randomize it, then it will have to be general. So the thing we're randomizing are the sizes. You can't assume that the first object or the second object is always going to be some range, some particular size. It's got to generalize across all of them. Okay, so what I'd like to do now, let's see, where am I at? I've got about half an hour, is that right?
More, a little bit more.
Okay, well, we'll see how it goes. So now what I want, so this is an example of what I meant by applying a particular mathematical lens, namely the lens of dynamical system theory, to a particular brain-body environment system, namely this relational categorizer, and working out an explanation as to what's going on in the operation of this system. How is it solving the task in terms of dynamical systems theory? Now what I want to do is show you the same kind of thing using the mathematical lens of information theory, looking at exactly the same passive agent initially, okay, but with this rather different lens. So let me just, as I did with dynamical systems theory, very briefly, because I don't have time to go through this in detail, but orient you to the kind of tools that we're going to be talking about. So the two main tools of information theory, which go back to Claude Shannon, are a measure of entropy, which basically is a number that you can assign to a random variable or a set of random variables that quantifies the uncertainty that you have about the outcome of making a measurement on that random variable. Okay? So if, for example, this random variable x always takes on the value 1, then you have no uncertainty, right? Because every single time you measure it, you know it's going to be 1. If, on the other hand, this random variable x is uniformly distributed across some range, then you have maximum uncertainty, because any possible value is equally likely. And entropy is just a way of quantifying that entire range of possibilities. A slightly higher order kind of measure that Shannon introduced is the mutual information between two random variables, x and y. And I think the most intuitive way of thinking about this is kind of a generalized measure of correlation. Standard correlation is only sensitive to linear relationships between variables, but mutual information is sensitive to nonlinear relationships between variables. Essentially, what it does in words is it quantifies the extent to which knowing the value of one random variable y reduces your uncertainty about another random variable x. Okay? So if these two are functionally related, then knowing y allows you to predict x uniquely. Okay? And that's the highest possible mutual information. Okay? If these two variables are completely independent, so the value of one has no relationship whatsoever to the value of the other, then the mutual information between those two variables is 0. And again, like entropy, mutual information is a way of quantifying the possibilities between those two extremes. Now, for what we want to do, this sort of standard two tools of information theory are not quite enough. One of the problems with mutual information is it just gives you a number. Okay? It gives you a quantity which says how correlated in general are these two variables. But what we're often interested in is the information over time. That is, the information that one neuron contains about something might be different at one point in time than it is at another point in time. So we have to actually unroll mutual information into a time-varying quantity. I think no, there it is. Okay. So we replace a number like this with a curve like this, and this number amounts to the average value of this curve. Okay? And technically, to do that, we have to replace random variables with random processes, which are time-indexed random variables, essentially. You have an independent random variable at every instant in time, and you're looking at the mutual information between each of those and some other quantity. It turns out that's not enough, though. Sometimes we need more detailed information about this informational relationship, and so we also use a quantity called specific information, which unrolls the mutual information along another dimension, which is it doesn't average over the stimulus possibilities, but rather accounts for each one separately. And what I've done here is I've unrolled it over time and over stimulus outcome. So any point on this surface says, at this time, the random variable contains this much mutual information about this particular value of x. So what we can see when we do this, I mean, if you look here, at any given time, any slice in time, so we stop unrolling over this, the output doesn't have uniform information about the different possibilities. It has more information about the smallest and largest values of x and less information about the intermediate values. Once again, this number corresponds to averaging over time and averaging across stimulus possibilities. So what we've done is take this number now and spread it out into this two-dimensional object, which gives us much finer information about the relationship between two random variables. And one final thing we have to do, because of this fact that this system changes over time, is we need to be able to have dynamic measures of information. And in particular, I'm going to assume a measure of information gain and information transfer. This actually turns out to be a really rich and contentious area of information theory right now, because doing these kinds of dynamic measures properly involves having ways to quantify mutual informational relationships between more than two variables. And so-called multivariate mutual information is very, very tricky to do right. And there have been different proposals that have been based, and there's a lot of controversy. So I'm actually using one that we developed called partial information decomposition. But I don't want to give you the impression that this is the standard way of doing it. This is standard. Everything above this line is standard. These two things are in the literature, so they're not things that we're proposing that are new and different. But these measures of information gain and transfer are things we're proposing. So they should be taken in the context of this is an ongoing controversy within information theory. Okay, so those are the tools we're going to use now, these various measures of information. So now what I want to do is revisit our passive agent first in the Q stage when we drop the first object, and then in the probe stage when we drop the second object. Okay? But using these tools now. So, for example, we can compute the time-varying mutual information between the size of the first object and the output of each of the sensory neurons. So that's what you're seeing here. Time is advancing this way. The sensory neurons are colored different ways. They're paired because of the symmetry in the task. And then you can see that information becomes available at different points in time as the object intersects the different rays. And then they have different profiles, and then eventually they drop off to 0. Because when the object is right in your face, all the rays are at their shortest possible distances, and you can't really distinguish anything anymore. Okay, it's like if somebody took an object and put it so close in front of your face that you couldn't actually see it. Right? The information that your brain is able to extract from that image goes way down if it's too far away or if it's too close, but when it's in an intermediate range, then you can extract your maximum information. So we can do this for all of the components of this system. This shows the mutual information profiles for each of the 3 interneurons. Okay, they start out with no information, and then as the sensors build up some information, that starts to get passed along to the interneurons. They start having information about the size of the first object, and I'll talk more about the particular profiles in a moment. And then just to kind of test your understanding, if we look at the motor neurons or the actual position of the agent, there's no information at all. Why? Yeah?
There's no possible decision yet, it's just the Q.
Well, that's correct, but the important other key thing is that these are passive agents. So they don't move during the Q stage. Okay? If they moved, then there's the possibility that they would move differently for smaller Q objects as compared to larger Q objects, and there could be some information at the output. But this is just a manifestation of the fact that this strategy is a passive strategy. So if you're going to stand stock still no matter what you see, then making a measurement of motor activity isn't going to tell you anything about what you're seeing. Does that make sense? The fourth sensor has no information because of the symmetry of the task.
So could you just remove it and have no?
Not for the active agents, but in the passive agents, yes. Except if you removed it after evolution, it could be that whatever value it takes on, even though it's constant, might be important for the rest of the network. You understand what I'm saying, right? So, but it does suggest if you hadn't put it in to begin with, it wouldn't matter for the passive agent and also for this passive agent. Okay, so there's one other thing else. So this is the kind of thing we can do with, this is not using specific information yet. This is just using time-varying mutual information. There's one other thing I want to draw your attention to before we move on from this slide. If you think about it, it's basically the point that you were making over there. You can't make your decision until you've seen the size of the second object. But the first object goes away after it reaches you. So somewhere in the system, there has to be some kind of memory. And I use memory in the sort of generic dynamical sense. There's got to be some state that's held somewhere in the system related to the size of the first object after the first object has disappeared. So it's clearly not in the motor system because there's no information. It's clearly not in the sensors because at the end, they all drop to 0 when the object disappears. It has to be in the interneurons. Well, let's look at this. Interneuron 1 and interneuron 2 have no information about the size of the first object at the end, even though they have substantial information at intermediate times. So it's only neuron 3, as Paul mentioned before, but now we're seeing this not through the lens of dynamical systems theory, but information theory, that has substantial mutual information about the size of the first object at the end of the trial. Everybody see that? Yes?
So am I right in thinking that it could perfectly work in N1?
Oh, yeah, absolutely. Again, absolutely. And it could also have been combinations of the interneurons. This is just this particular agent. It's N3. But that's not general. What is general is you could do this kind of analysis on any of the evolved agents and look at the profile, the informational profile.
And at least one of them, or some combination.
Something has to, right? Yeah.
And why are those ones not symmetrical?
Which ones?
Interneurons.
Because they're fully coupled, right? I mean, so, and there's no symmetry imposed on the parameters of the evolution, right? So,
The weightings are different.
yeah, right. The weights can be different. The biases, the time constants, they can all be different. So but I do want to emphasize to people, because it's easy if you're not used to looking at information theoretic plots, don't think of these as neural activities, because they're not. What I'm plotting here is information. Okay? So just because something is flat, like here, doesn't mean that the motor neurons aren't activated. Another way to stay in place would be to activate your left and right motor neuron full on, right? And they would be opposing one another, and therefore you wouldn't move. But informationally, there's no information content in doing that, because it doesn't vary with the size of the object. Does that make sense? So don't keep stopping yourself from slipping into viewing these as interneuron or neuron activations. They're different now. They're informational things.
But there's one thing that confused me. So you're emphasizing this point of memory now, right? But
Yeah.
given the lateral coupling among these what you call interneurons, that's a substrate of the memory.
In this case, yes.
Right? So in this case.
In this case.
So why do you emphasize this memory component so much at this point?
I'm only emphasizing the fact that one of the questions you might like to answer by this informational analysis is where in the system is the information about the size of the first object, given that you need it for the second half of the problem, right? Okay, so let me just go into a little bit more detail. So let's focus now just on that third, N3, the third interneuron, because that's the one that has all the information. So I'm just re-showing you its profile. Now we can use specific information to unroll that picture across stimulus values, so across different sizes of the Q object. And from that, we can compute, for example, measures of information gain. So we can see that neuron 3 gains information about the size of the first object in waves. And it's non-uniform across the different sizes. So for example, look at this part right here. This wave first applies at large object sizes and then doesn't actually cover smaller object sizes until later. So there's a particular profile across time and stimulus values, in this case the size variable. There's a particular profile in how neuron 3 gains information. And by looking at the information transfer, we can actually make sense of that. So this shows you the specific information transfer from each pair of sensory neurons. So this transfer comes from this pair of sensory neurons, and that accounts for these two waves of gain. This information transfer accounts for this wave of gain, and this information transfer accounts for most of this part of the gain. Now, one thing I'm not showing here, you might conclude that informationally speaking, interneurons 1 and 2 must not do anything either, because their information ultimately falls away. But the fact is that the interactions between neurons 1 and 2 with neuron 3 are important. And there are small features of this information gain plot that can't be explained just by transfer of information from the sensors. If that were true, if you could explain all the information gain by the sensors, then this would be a purely feed-forward effect. but it's not. There's also some lateral information transfer. It's very small in this particular circuit, but in general it can be much larger, and that involves interchanging of information among the interneurons. Okay. All right, now I want to talk about the probe stage. So now we have a story, an informational story about what's going on. We can track when information about the Q object becomes available in the sensors, when it becomes available in the interneurons, and which interneuron sort of holds the memory for later processing. And we can also unroll that and go into fine details about where different bits of the information come from within this circuit. Now I want to switch to the probe stage, and I could repeat the same analysis as I did with the Q size on the probe size, the size of the second object. That's a perfectly well-defined information theoretic question. I could do similar plots that I just did. But if you think about it, what we really care about for categorization in this task is the relative sizes of the two objects. So that's a higher order information variable, which we can just define. It's a binary random variable that takes on the value 0 whenever the probe is bigger and takes on the value 1 whenever the probe is smaller. Okay? We can equally well analyze where and when information about relative object size occurs in this system. So again, just for reference, I'm going to put that back up. As I hope you'll see why in a minute, I'm going to start at the motor end and work back. So here is the mutual information between relative object size and x, which is the position of the agent's body. Why do I start there? Because in the end, the agent's decision is expressed by its position. It's expressed by the way it moves. So what you're seeing here is that there's no information about relative object size in the agent's position for a while, but then there's a point at which suddenly there's a sharp transition in information, and then it kind of flattens out, curls up a little bit more. But this is the important event right here. That sets a kind of time frame within which the agent's decision is expressed. Now, if we want to figure out where that decision came from, information theoretically speaking, what we have to do is look at what are the possible sources of that information that came into X. So the most obvious ones are the motor neurons. Okay? So if we look at when do the motor neurons have relative size information, again, we see early on they have none, but then eventually one of them, the right motor neuron in this case, shoots up, whereas the left motor neuron doesn't really show any information about the relative object size until much later. Now again, I want to stress, this does not mean that the right motor neuron turns on. In fact, it turns out that the right motor neuron turns off proportional to the information about the relative size. So this is an informational picture. Okay? And you might hypothesize that because this increase in information precedes this increase in information, whereas this one follows it, that this may be the source of the information that ultimately is expressed in the agent's motion.
So Randy, what is the max information? Just to understand the numbers on the
These are normalized
right.
by the entropy of the random variable that we're talking about. So in this case, they're normalized by the entropy of the relative object size. They're normalized to 1.
Okay, very good.
Yeah. Okay. So once again, we can backtrack further and ask, okay, where did the motor neuron, the right motor neuron, get its information from? Okay. So here are the pictures of the relative object size information in the 3 interneurons. And once again, if we want to see what happened here that caused this to suddenly jump up, well, this one wasn't changing, this one wasn't changing much, this one changed a lot. So we might hypothesize that interneuron 1 increase in information was responsible for the right motor neuron's increase in information, which was ultimately responsible for the movement that reflected the decision. Now, one thing you have to be careful about here is when the agent starts to move, that changes what it sees, which changes its interneuron activations, which change its motor neurons. So I'm giving you a kind of semi-linear, in the sense of this thing, then this thing, then this thing, story of what is really a feedback system, which is why dynamical systems tools are so appropriate. Okay. Nevertheless, I can use the information transfer measures to show that, in fact, the information transfer peaks. So in this case, between neuron 1 and the right motor neuron, peaks right around the time this is happening. And this information transfer from here to here peaks around the time this is happening. Okay? So one thing that information theory alone, this is very important, cannot tell you is causality. Information is a way to quantify correlations. On the other hand, if you have access to the architecture of the system, which we do, and if you have access to the causal mechanisms of the system, which we do, then correlational hypotheses about what causes what can be turned into causal stories about what causes what by investigating the actual underlying mechanisms. And so the story I'm telling you informationally can actually be grounded causally in the underlying system. Now, everything I've said so far has been about passive agents. I told you I would say something about active agents, and that's what I'm going to do now. Yeah?
Just before you move on to the active agents, so if I remember rightly, during the Q stage, wasn't it the opposite interneuron?
It was neuron 3 that played the important role in remembering, quote unquote, Q size information.
Right. Okay.
But it's N1 that actually seems to have the transition of relative object size information. By the way, nobody called me on this, but it's fun, so I'm going to do it anyway. Did anybody notice anything funny about neuron 3's relative size information here? Because you raised, right? Neuron 3 was the important one at the beginning.
Yeah. I've got the graph here in your paper.
So it's right here. So neuron 3 begins the probe stage with relative size information. Now that's a little puzzling, but maybe it instantly sort of figures things out. What I don't have the plot for, but if I actually show you the relative size information in N3 before the probe object is even dropped, it's non-zero. Think about that for a minute. Neuron 3 has an opinion, right, basically, about the relative object sizes before it's ever seen the second object. What is this, a precognitive agent?
It has an accurate opinion, in fact.
It's not super accurate, but it's somewhat accurate, yeah. I mean, what is this, a precognitive agent? Can it predict the future? No. What it's doing is the following. A detail that was up there, but I didn't emphasize, is there's a particular range over which the sizes from the objects are drawn from. So if the Q object is very, very small, the chances that the probe object will be smaller are very low. If the Q object is very large, then the chances that the probe object will be smaller is much higher. So based on the size of the first object it sees, N3 seems to carry predictive information about what the decision will be. But interestingly, that's not really used by the rest of the system. It's just there. It's the kind of thing that you see all the time when you do these information theoretic measures. Depending on the questions you ask about your system, you find all kinds of information about different things in these systems.
But there are two. Is it not just expressing the mean size of the objects or the stimuli you've been using?
It's expressing a little bit more than that. It's expressing this consequence that I said, that depending on the size of the first object, you have a higher or lower probability of the second one being smaller.
Well, but have you confirmed that by then analyzing the sequence which you have presented yourself?
The sequence that you presented.
Because you're saying now, given stimulus 1, I have a prediction about stimulus 2.
Yes.
Right? So that would mean that there there's some history about the sequence.
I mean, that is the analysis. That's showing that if you make a measurement, well, actually.
No, but what I was asking for, how can you prove to me it's more than just the mean size? You said, no, it's more than that. Right?
I mean, I'm not sure how you would, so if you looked at, if I defined another random variable, which was the mean size, you would also find all of these things carry information about that. So, I mean, in that sense, you can't resolve the question, because the way information theory works is you come to a system with a set of questions, with a set of quantities that you're interested in, and you compute the informational measures of those quantities.
Okay.
So you could certainly measure other things, and you'll also find some profile.
But on top of that, this answer shows us something about the dynamics of the model, right? Because apparently at time 0, so at the start of the trial, there is already a non-zero state in these units.
Sure.
Apparently.
Well, because
there's no reset.
No, but wait, there's no reset between between the probe and the Q, or the Q and the probe. Yeah. If there was, you couldn't solve the problem.
Right, exactly.
Right?
Just to emphasize that.
Yeah. Okay. Absolutely. Yes.
Is there some assumptions about the distribution of the sizes?
Yeah, they're uniform.
So how does it know about where does it know this information?
Yeah. So how does this N3 in the beginning,
Right.
non-zero, where does this information about uniform distribution get transmitted?
I mean, whenever you compute these information theoretic quantities, what underlies their computation are probability distributions. So the uniformity of the Q and probe sizes enter in as the distributions for those two things. During evolution, this agent only ever saw a uniform distribution. So if you had imposed some sort of bias distribution over it, and it evolved to deal with that, then I would expect that you would see biases to its judgments as well. Does that make sense? Yeah. Okay. All right, so now what I'd like to do is talk about the active agents, and then I may have to start wrapping up, so I may not get to the connection between the two. Okay, so I mentioned that there are these other agents that move throughout both the Q stage and the probe stage. And we can analyze those agents dynamically and information theoretically. I'm just going to do them information theoretically. So I'm going to kind of repeat very quickly the same set of analyses we just went through in more detail. For the passive agents, I'm going to repeat them on the active agents. So let's first look at information about the size of the Q object in the 3 interneurons. Okay, hopefully you'll know right what to look for in these pictures. What's puzzling and concerning about these pictures? There's no information in interneuron 1, interneuron 2, or interneuron 3 about the size of the first object. I'm not showing you here, but the sensors work the same way. So there's no information about the size of the first object in the sensors either. Okay? So how is it, then, that they can do a comparison between the size of the first object and the size of the second object if they have no information in the nervous system?
In the position.
Yes. If I look at the information, the mutual information between the size of the first object and the agent's position, I see, after a short delay, very strong information. So unlike in the passive agents, what this active agent does is it sees the first object and it moves an amount that's related to the size of that first object. So it's holding the memory, if you will, not in its nervous system, but in the position of its body. Okay, and that has a name. It's called information offloading. It's the same thing you do when you arrange your desk in such a way that the higher priority items are closer to you or something like that. Okay, you don't have to remember what's more important because you've laid things out in your relationship between your body and your environment to store that information. It's the same thing you do when you write things in your calendar.
But that would only be true if there's a possibility for the agent to recover its position relative to some initial position. Right? If it would have no ability to access X, then there is no memory. Right? So this only works as a memory when it has access to the information.
It doesn't have access. So what it has, what this relies on, is in fact that the objects fall from the same place. For instance.
Exactly.
Yeah. Yeah. But I mean, the information offloading is a general property. The information offloading in this agent is a fairly simple trick, is what you're saying.
Because there's still a reference.
That's right. Yeah.
That's my only point.
Okay. The next thing I want to point out to you is during the probe stage, when we drop the second object and we look at the information that this is sensor 1, but this would be the same for all sensors. The information that the second object has about, the information that each sensor has about the size of the second object. Again, this should be worrying to you. That's flat. There's no information in the sensors about the size of the second object. But what there is, so I mean, how is probe size extracted and used in order to make this comparison? Well, what we do find is that there is what's called synergistic information between the sensory output and the object size. So if you take both of them into account together, then you do find non-zero information about the size of the second object. And there are these measures, which again fall into that controversial set. There are different ways in information theory to measure synergy, to measure the combined effects of multiple things. What information you get out of measuring a set that you can't get out of just looking at the individual components in the set. That's what's quantified by synergy. And this is an example of making use of that. And the way it works is, remember, you move some distances proportional to the size of the first object, and then what you do is you start moving back towards the midline again. And for smaller objects, you encounter them earlier. For larger objects, you encounter them earlier than you would encounter smaller objects. So this is the way it actually integrates the information that's in the position of the body with the falling second object. So in a sense, what it's actually done, and this has a name too, it's called information self-structuring. It's basically offloaded not just the memory, but the decision-making itself has all been offloaded to the relationship between its body and the environment. It set things up so it can basically directly perceive the relative size of the second object.
But now this is an aspect of the agent generally highlighted previously, right? So before we all looked at moving left or right, to
right.
move away from the stimulus.
Right.
But apparently there's also a fixed tendency to go back to the center point.
Only in these active agents, not in general.
No, for the active agent.
And only in this one can I confirm that, because each active agent has a slightly different strategy, right? So again, the details will differ. Yeah. Other questions?
So that would mean that you also have a certain fraction of agents that actually fail in performing the task, because they might not have this tendency to move back to the release point.
So, I mean, I don't have the statistics. Usually, about half of the agents that work are passive, and about half of them are active. But I don't remember how many of the runs we did actually work, but it was pretty high, something like 70 or 80%. This is not that hard of a task. Some of the tasks are much harder than this one. But it's rich, because there are lots of interesting layers that we can explore using dynamical systems and information theory, which is why I focused on it here. Okay, I'm going to skip, unfortunately, this bit because it's somewhat detailed. But I hope to at least have piqued your interest. And there is a paper, as someone mentioned, where we look at this in detail. That I've given you a story dynamically in terms of transients and these moving manifolds and equilibrium points and bifurcations. I've given you a dynamical system story about how a passive agent works. And I've given you an information theoretic story about, well, there's information about this here, and later there's information about that there, and you can see the transfers going on and so on. Obviously, it would be interesting to see how the two fit together. What's the relationship between that bifurcation that you see in the dynamical systems story to, say, some abrupt change in information that you see in the information theoretic story. That's the kind of thing that we want to try to do. Okay. What I'm going to do instead is just wrap up with 2 more slides, and then I will be done. So to summarize, what I've tried to do here is the following. I've wanted to suggest to you this sort of methodology as an interesting one that you can use evolutionary well, in general, I guess, let me break it into 2 pieces. In general, I've suggested the idea that toy models, in the physics sense of toy models, can be very useful for building our intuitions and testing out our ideas and evaluating and developing new tools and so on for problems in the behavioral and brain sciences. I've tried to suggest to you that the particular way that we do this, namely using evolutionary algorithms to generate model brain-body environment systems, and then treating them seriously, actually studying how these things work. Okay, not just, oh, we did it, great, let's do something else, but actually treating them as worthy objects of analysis, almost as if they were biological systems. And I've shown you, we've looked at one particular example, this relational categorization task. We've gone through a dynamical explanation and an informational explanation of that task, and I didn't get to this part, the relationship between the two. Okay? And the last thing I wanted to mention is just everything I've done here is just one example of this approach. So there are many, many other examples where we've evolved and subsequently analyzed various brain-body environment system models. And the one I guess I want to end with, because I think that may tie in here, is something that we're especially interested in, is applying exactly these same techniques to problems in C. elegans. So here we're actually evolving and analyzing models that are based in the biomechanics of C. elegans' body model, the connectomics of bits and pieces of its nervous system, hypotheses, some grounded in data, some not, because of limitations that we currently have in doing neurophysiology in C. elegans, hypotheses about neurophysiological mechanisms, and then the parts that we really don't know anything about, we use evolutionary algorithms to try to fill in the details in such a way that the model C. elegans produces some behavior. If you do that once, it's not very interesting, because there's no real reason why what the evolutionary algorithm came up with has any bearing with the biology. But if you do it thousands of times, you start to explore the space of possibilities that I mentioned earlier. And then you can ask interesting questions like, if I ran the evolutionary algorithm 1,000 times, maybe 70% of them worked. If I look at the different solutions that they get, they fall into maybe 4 or 5 different classes. This class of solutions has the following property. If I do something to it, if I reverse the gradient, the chemical gradient, if I knock out a neuron or whatever, then all of those evolved models have a certain deficit. What's the corresponding effect of that same manipulation in the animal? That can be used to either lend evidence for or eliminate a whole class of possibilities, which they can then feed back into a future evolutionary run to disallow or encourage that particular feature, depending upon the results of the experiment. So I think it lets us feed into this modeling experiment loop much earlier than if we adopt the attitude of at least some neuroscientists that I've spoken to about this, that, oh, you don't know enough to model yet. You have to wait till the experiments are done and you get all your data, and then it's time to actually build a model. All right, anyway, I think that's all I'm going to cover for today, so thank you for your attention.
Well, thank you very much, Randy. That's fantastic. Questions, please?
Here, I was just wondering, in terms of the passive agents, in the motor neurons you said, and the example you showed, there was no information about the relative size, the size of the cube.
The size of the cube. There certainly is information about the probe and the relative size. I showed the relative.
Yeah, exactly.
Yeah.
But in principle, even if I'm a passive agent, I can ramp up the activity in both neurons, as you said, to somehow code this information, so I could have information
could.
in the motor neuron. Have you seen any that would do this?
There's one I can think of that had a little bit of information. It did exactly what you said. They were balanced, but the overall level went up or down a little bit based on it. I mean, there's really not much. I mean, what defines as you know, a passive agent is that they don't move. And it's very hard to keep the motor neurons balanced when you have asymmetries in the interneuron layer, right? And so usually not moving means that they're just kept pegged on or off, essentially. But yes, I did see one with a little bit of information. You're right. In theory, it's certainly a possibility.
So I thought this your whole account and analysis was it's inspiring, really, and immensely intellectually satisfying, especially to see the richness that's required. But it's also actually ever so slightly depressing in the sense that you're thinking, how on earth is this kind of analysis going to scale up to to a bee, say 900,000 neurons, let alone sophisticated cognition of other animals.
Yeah. So this if I had to predict, like neuron 3, one question that I almost always get, it's that one. No. But I mean, it's always a good question.
Randall has a neuron 3 as well.
It's always a good question. So I guess there are a couple of things. First, about the depressing character. I prefer to think of it as realistic. I think anybody who thinks that somehow it gets a lot simpler when you have 100 billion neurons than it is when you have 3 is just deluding themselves. I really think what's interesting about these models is that you get to play with these issues that are going to be important at the larger scale. You just don't know it yet because you don't have access to the level of details and the range of different situations that we can put these things in order to probe them. Now, I could be wrong about that. It certainly could be that it all simplifies when you have these big things. And there are statistical arguments you can make, and you can draw on analogies to statistical mechanics and physics and so on. But I think there are good counter-arguments to that. But so I prefer to think of it as realistic rather than depressing. It shows, it's a reminder to stay modest about what it is that you can actually accomplish and how simple your story ends up being. The scaling up issue, though, is a slightly different issue. So another lesson that some people take from this work is, oh, well, you did this beautiful analysis, but you couldn't possibly do it at any interesting scale. It's only dependent on the fact that you have 3 neurons or whatever. That's one of the reasons why I'm interested in studying C. elegans. So it's not 90,000, it's 302 in the hermaphrodite. But it's certainly an order of magnitude bigger than anything that we've looked at so far. And so I think the proof is in the pudding. I mean, you've actually got to see how you can deal with that. Clearly, the mathematical techniques of dynamical systems theory and information theory are not scale dependent. The actual techniques themselves, it's not like the math falls apart when you go to higher dimensions, okay, in any fundamental way. But what does become more and more difficult is drawing the kinds of pictures that I just drew for you and walking you through the intuitions graphically. Not because of any limitation in the math, but in our limitation, that we can't actually visualize these higher dimensional spaces. And so that continues to be a real problem. And you're constantly on the lookout for things like Paul mentioned, where you get this, in this case it was a modest one, but this 3-dimensional state space collapses into a 1-dimensional curve under certain conditions. Okay? That kind of stuff probably happens all the time. If you think of all the degrees of freedom you have in your body, it's very, very high. But when you're doing particular tasks, it tends to collapse to much lower degrees of freedom. And if you're interested in focusing just on the context of some behavior rather than everything you do in your life, then there's probably some lower dimensional description that you can analyze. And the variables of that description can be the state, the space that you do your dynamical analysis in, and they can be the random variables over which you do your information theoretic analyses. So I think, again, realism, and I think there are steps forward, but it's hard. It is hard, and that's the concern.
Hi. So my understanding, perhaps naive, is that there's a relationship between information theory and dynamic systems.
Oh yes, that's the whole part of the talk I didn't give. But yes, there is.
But that's expressed when you come to chaos through things like Lyapunov exponents, things. So could you comment on where chaos lives in these systems?
Well, I mean, you don't. So the relationship between dynamics and information theory doesn't depend on chaos. But it is true. I mean, there's a perfectly interesting and well-defined relationship in these systems, which are non-chaotic. But you are right that chaos is where people have tended to focus most. And that is because you have this very natural uncertainty relationship, that you don't know the state to an infinite precision. Therefore, it behaves as if it's a kind of random variable. And you can ask questions like, how does my ability to predict the future, given a measurement at some finite scale now, how does that decay as time passes? So it's a very natural place to marry these two things. But there's a much broader connection between the two than what we've seen in chaos so far, I believe.
So, Randy, let's see if I can pique your neuron 3 again with this.
Okay.
Okay, so so in some of you're really advancing this toolbox we have to, if you want, dissect the information transfer going on in biological systems or biologically like systems. But on
Was
the
that a
other hand,
question or a statement?
no, that's my the assumption that,
Okay,
that drives
because
my-
that's one of the things I'm trying to do, but I hope not the only thing. But
no,
anyway.
wait, but that's the one I want to
Okay.
focus on. On the other hand, she also made it very clear, also in the title of the paper we see there, it's brain-body environment systems.
Yes.
But in the whole analysis you gave us, body and environment was actually removed.
No,
Or let's say
not for the active
strongly
agents.
reduced.
Not for the active agents. It's simple. You're right, it's
Well,
simple.
the morphology of the body was only expressed as position in space of sensors.
Yes.
That's not a body. That's a position in space of a sensor.
There are non-neuronal state variables in the system. That's what
No,
I
wait,
mean
but
here.
I would like to hear your comment on this, right?
Yep.
So under what conditions do you think we can really start to bring in a more realistic sense of body and environment in this analysis, or would that require a completely different approach?
No, I mean, there's nothing in what I've said that the body state had to be a single variable, which was position. It could have been more states in the body than there were in the nervous system. I mean, in the C. elegans stuff we're doing, one of the things that we're doing is working with body models that are I think about 150 dimensional is the current thing that we're looking at. So I mean, bodies can be quite complex things. Environments can be quite complex things. I have tended, the one thing I would agree with in the statement that you made is that I have tended to focus on the neural stuff. And the body and environment stuff has tended to just be minimal, but there. The key thing is that you get stuff like this information offloading just by having that one feedback with body position is all it takes to start getting active perception, to start getting information offloading and information self-structuring and all these kinds of things, to start coming face to face with non-autonomous transient dynamics as compared to attractor-only dynamics. All those things already raised by just having that single feedback loop.
Right.
And so that's why I focused a lot on that. But don't misread me as suggesting that therefore the body is and the environment is trivial. It's a matter of, sort of conservation of complexity. You can only handle so much complexity, and where do you put it, and what questions are you... I mean, part of the thing is I often give these talks on this stuff to neuroscience audiences, where it's even more effective. If I had a really complicated body and it had some impact, they'd go ho-hum. But if you have a really trivial body where you can actually point to this one single feedback and yet all these new effects come up, and it starts to be an invalid question to ask where in the nervous system is the memory stored, because it's not in the nervous system at all, that's actually a very effective way of demonstrating it. So.
Randy, so you mentioned active perception there. So one thing that struck me from the talk is that, hey, you've got a system where you can start to analyze the trade-off between active and passive sensing. And so you could imagine, in this case, there isn't really much choice. Either strategy works perhaps equally well, and that's why you get
That roughly
50%
seems to
of
be the
each.
case, yeah, but it's certainly not
Yeah.
always the case.
But so it would be interesting maybe to look at variations of the task and to see which ones favor passive and which ones favor active sensing approaches.
There's an earlier attribute categorization or classification. Again, I don't care what term you use, but task where I used a similar agent and you dropped either circles or diamonds, and it had to discriminate between which are the circles and which are the diamonds. I think it caught the circles and avoided the diamonds. And there, it always used active perception. And the reason had to do with the fact that the difference between the circles and the diamonds was very small relative to the spacing of the ray sensors. And so by moving the rays back and forth multiple times across the object, it could get a higher acuity discrimination. And so we were able to show in that case, you always got the scanning behavior. In here, as you point out, it's not really necessary because the fact that the object itself is falling over time gives you a temporal sample of things. And there's not really any need to move.
So that even in these simple models, you may be able to draw some general conclusions about when we see different strategies in animals, why they're doing this.
Yeah, I think one has to be very careful about the kind of conclusions one draws from these sorts of models. So one of the distinctions I make between this kind of work and the C. elegans work we're doing, for example, is that one of them is actually geared towards making experimentally testable predictions. Okay? This really isn't. I'm not making any claim about human categorization. And that's why I use this term from physics, toy models. Toy models can be completely wrong. Okay? 2 + 1 quantum gravity is a toy model that's used to look at quantum gravity in physics. It gets really fundamental features of the universe wrong. Okay? But it's a very important tool for working out what it means to combine general relativity and quantum mechanics. I think it's important to separate out these two classes of models. And this class of models is about, as Paul said, tool development, but also, I think, teaching us collectively about, for example, the consequences of feedback through the body and through the environment. It's absolutely fundamental. It completely changes the way you have to think about what a nervous system is and what it does. And that kind of thing, I think, can be also illustrated here. If that leads someone to go off and do a different experiment or think about their system in a different way, I consider that a success of a toy model. But I'd be very concerned about concluding, because in my particular setup, I see 75% active and 25% passive, that says anything about the relative benefits for a biological system. So I just like to be really careful.
Well, but that's contradicting almost the language you're using here. You're talking about minimally cognitive behavior. You're talking about perception of affordances, selective attention, short-term memory. So these are psychological concepts.
The reason why you do that is if I came in here and said, look, this florgle has a boogle that wiggles, okay? It wouldn't mean anything. The only way you can communicate concepts is by using vocabulary that people are familiar with, okay? Each of those terms I actually, like minimally cognitive, has a very specific meaning, which I laid out when I first defined that notion. It doesn't mean that these are just little people or anything like that, okay? It has a very specific meaning, which I can elaborate, but the point is, the more general point, is that is a pedagogical move so that you can communicate with people. I have a little story about that. You mentioned my dissertation. What I did in my dissertation was basically build in the computer, a model cockroach. Legs, body, nervous system, everything. Very simple. I tried to get so one of the things I worked on was insect walking. And initially, I had a round body with 6 legs, and it modeled that body as a Newtonian mass with collision and everything. I brought in some insect walking people. They saw this circle walking across the screen and said, okay? I replaced that circle with the canonical now image of the artificial insect, which is basically modeled less realistically in respect to collisions. But it looks like an insect. It's got a head, it's got antennae, it's got circi. And I was actually invited to give a keynote at the neuroethology meeting about that work way back, okay? So you don't underestimate the importance of being able to engage people with the vocabulary and with the images that they respond to. On the other hand, don't believe your own PR. That just because I did that, somehow it's more insect-like than when I had the circle. So, little soapbox, but I think it's an important point.
Sorry.
So given your answers, I somehow get the idea that also you hint to the C. elegans that these methods are applicable once I would know all the internals of the brain and all the neurons. But in principle, I would say you could also apply it to fMRI or to much more broader information that you can get.
So I don't understand the first point. The method of evolutionary algorithms is applicable precisely before you have all the... If you had a complete database of all the biophysics of C. elegans, you wouldn't need to use evolutionary algorithms. You would still need to do these analysis techniques. So let's be careful as to which of this method that you're talking about.
I was more applying now to the information theory or the dynamic analysis.
So information theory, for example, unlike the dynamical system stuff I did, doesn't require everything. What it requires is just the ability to measure some variables and then compute these sort of correlational relationships between them. So information theory is a very hot area in neuroscience right now. And it doesn't depend on having all the biophysical details, or it wouldn't be nearly as hot as it is. Dynamical systems theory is a little more difficult. You need to have a state space, which is not as trivial as you might think. A state space has the property that there exist some dynamical laws such that given that state, the evolution of the system is deterministic. Okay? So that doesn't mean you have to have everything, but you have to have a level of information at which you can formulate laws at that level where the state you have is sufficient to determine the future action of those laws. Sorry, I lost what you were getting at, I guess.
Applicability. So Armin was saying it has a broader application.
Oh, yes. Yes, so I mean, dynamical systems theory, you certainly don't just apply it like this, right? I mean, you can do it at a very high level. I mean, people do dynamics of, as you said, you can do fMRI patterns, you can do PDE type analysis of pattern formation in brain activity. You can do, there's lots of stuff in complex networks where you're looking at synchronization, for example, occurring across brain networks, things like that. That's all dynamical systems theory. That's why I made that distinction earlier about dynamical systems theory, which is a body of mathematics and sort of theories that you're putting forward about how some particular system works.
So I wanted to just come back to the comment that,
Sure.
that you think these systems are too simple to help us understand these higher level phenomena. So
No, I didn't say
but
they're
you're
too simple.
being very careful about it. So you're
Yeah.
using the language, but your explanation is that this is to engage people. But I think maybe you really believe actually when you take this task, the characteristics of the short-term memory problem here do reflect some interesting properties of short-term memory generally that animals have to face.
I think this issue of offloading versus internal storage is interesting and likely to be very relevant to the breadth of memory possibilities in human beings. But I wouldn't go any further than that,
Right.
I guess. So yeah, I am trying to be a little bit careful. I mean, the problem I find is that if you're not careful, models can be very powerful. I mean, they're like acid. They can sort of eat through anything. And you can convince a lot of people of things by throwing up a model and doing some math or making some pictures and so on. And I think it behooves the people like us that do these models to be very, very careful about what we do, what we are claiming, and what we're not claiming with any particular model. Because every model does violence, right, to the actual phenomenon that it's studying. It cuts away 99% of what's going on to focus on just certain things. So there are bits of it that you want to put forward as this is what I'm really claiming. And lots of the other things you don't want anybody to read anything into. They're just arbitrary decisions that you've made in formulating this particular model.
I think one of the things that I get from your work, which I think was made very explicit by Valentino Breitenberg in his Vehicles book, that when we look at biological systems, there's a real tendency to overestimate their complexity. And Breitenberg, as a neuroscientist, made that point with, in some ways, very simple theoretical models that he did sort of thought experiments with. And one of the great advantages for me of the simulation approach, and generally of the synthetic approach, is that you can ask, well, how complex are these problems that we're looking at? Are our intuitions about something being difficult wrong, or our intuition is about something being easy wrong. And we can find that out through these synthetic approaches.
Yeah, but just because I can generate complexity from simplicity doesn't mean that every complex system is at root simple, right? I mean, the converse doesn't follow from that. It could be. It certainly could be, but it doesn't. You can't follow. That's an independent thing. I happen to believe biological systems are complex, and we have a long ways to go to develop. I mean, there's a part of this talk I cut out, which I was going to. It's a good thing I did because I ran out of time anyway. But sort of arguing about the need for certain kinds of theory development in the behavioral and brain sciences more generally that just hasn't been done and hasn't been done well. There's a need for that kind of thing, but we really haven't made a lot of progress yet on that.
Okay, well, I think maybe we should stop there for a break, and we'll start again at midday. So once again, thanks very much, Ryan.